When it comes to muscles and muscle tissue, there are five main functions: to produce movement, to maintain posture, to stabilize joints, to generate heat, and to protect organs. Muscle tissue has four different characteristics. Muscle tissue has excitability, which means that it's able to receive and respond to stimuli from the nervous system. Contractibility means that it has the ability to shorten, which is the main function of how muscles work. Extensibility, meaning muscles can stretch. And elasticity, returning to the resting length after being stretched. There are three types of muscle and muscle tissue in the body. There's skeletal muscle tissue, smooth muscle tissue, and cardiac muscle tissue. Those types of muscle tissue differ in the structure of the cells, the function of the muscle itself, the location where it's found in the body, and the activation, how it gets the signal to contract in the first place. So in this unit, we're going to talk a lot about skeletal muscle. We'll also talk some about smooth muscle. Cardiac muscle, we're not really going to address very much in this unit. We'll talk much more about that when we talk about the cardiovascular system in the heart. So skeletal muscle. Skeletal muscle, the cells that make it up are called muscle fibers. So muscle fiber, skeletal muscle cell, they mean the same thing. So skeletal muscle fibers have a very unique structure. They are extremely long cells. While they're still microscopic, they are much longer than what you would consider an average cell to be. They are cylindrical. Because of their long size, they have multiple nuclei to control the larger cell. It's striated or striped and contains multiple mitochondria so that it has the ability to produce lots of energy. The function, they are under conscious control, meaning they are voluntary. They contract rapidly, so a very fast contraction rate, but they also tire easily and they must have a rest period to regain their full function. So this is a microscopic image of skeletal muscle cells or skeletal muscle fibers. Each of those sort of parallel sorts of lines, each of those are muscle cells. And so you can see they all pretty much extend off of the image because they're so long. You can see nuclei in the cells, and there's multiple nuclei in each cell. And then you also see the striations or the stripes. So we're going to talk about the organization of muscle tissue. So skeletal muscle fibers have different connective tissue wrappings, and the wrappings end up creating these different sorts of structures, which then allow the muscle to do different functions. So each and every muscle fiber has its own connective tissue wrapping called the endomysium. Muscle fibers then are grouped together and wrapped in connective tissue wrapping called the perimysium. And that bundle of muscle fibers wrapped in perimysium is called a fascicle. Fascicles are the functional group of muscle fibers. The direction of fascicles in a muscle will determine how it contracts. And then finally, the entire muscle, all of those individual fascicles are wrapped with epimysium. So that wraps the entire muscle, and that fuses into a tendon, which the tendon then will attach to a bone, or it can fuse directly into the periosteum of a bone. So this is just reiterating the structural components of muscle and the different wrappings that each has. There are two different ways that skeletal muscles can be attached to bone. Now, first of all, most muscles will span joints and attach to bone in two different places. Because it's spanning a joint and attaching in two different locations, when the muscle contracts, it's able to pull on a particular bone. So the insertion is the muscle attachment on the movable bone. When the joint is used, the bone that moves, that muscle attachment is the insertion. The origin is the attachment of the muscle that's on an immovable bone. So these are the two ways that the muscles can be attached. They could be directly attached, which is when the epimysium is fused to the periosteum. So this would be like the intercostal muscles of the rib cage. Or what we more normally think of is the indirect attachment, where that epimysium, the connective tissue wrapping, extends and kind of comes together, forms a tendon, and then the tendon is what attaches to the bone. Back in biology, you learned about a generalized cell and you learned about the different organelles. But muscle fibers, they have all those usual organelles, but then they also have some specialized organelles, or amounts of organelles that you wouldn't expect in a typical cell. Like we mentioned before, muscle cells will have multiple nuclei, and that's because these are large cells that need more control mechanisms. They have multiple mitochondria because they have very high energy requirements, so need more of those powerhouses to produce the energy. And they have multiple myofibrils. Now, myofibril is an organelle we did not learn about back in biology, and that's because myofibrils are only found in muscle fibers. They are contractile organelles, and so it's the myofibrils that allow muscle fibers to be able to contract and cause a muscle to shorten. So they're unique to muscle fibers. So myofibril structure. A sarcomere is repeating units on a myofibril. So the myofibril will have these units called sarcomeres along its entire length. The sarcomeres are separated and linked together by Z lines or Z discs. So you can see the picture up here is a muscle fiber. And then what they've done is enlarged a myofibril. And the myofibril here, we can see there's one complete sarcomere, which is enclosed by those Z lines. And then there's another sarcomere off to that side and another sarcomere off to that side. Myofibrils also contain myofilaments. There's three main myofilaments in a myofibril. There are myosin myofilaments. These are thick filaments that we find in the middle of the sarcomere. So think myosin is found in the middle. Actin are the thin filaments that will always be attached to the Z lines. And titin is an elastic recoil filament. It stabilizes and centers the thick myosin filament. And all of these myofilaments are composed of protein, which is why muscle has such a high protein content. A sarcomere also will have specific banding patterns, and the banding patterns, some will change depending on whether the muscle is contracted or relaxed. So the A band is the entire length of the myosin. And so the A band will never change in length because the myosin will always be the same length. The H zone is the A band where the myosin and actin do not overlap. This disappears during contraction because the actin and the myosin during a contraction do end up overlapping. And then finally, the I band is the actin connection to the Z lines where there's no myosin overlap. And that too will shorten during contraction. So if we look at the picture, the A band is the length of the myosin. The H zone is this middle section here where we have no overlap. We have only myosin and no actin. And then over here, the I band is where we have no overlap. We have only actin, but no myosin. And so during contraction, when that sarcomere ends up shortening, we end up losing the H zone because the actin comes in to overlap, and we end up losing the I band because the myosin will start to overlap. So here's just another image of those same things. Here's a microscopic picture of the different banding. So muscles will contract by the sliding filament model of contraction. And this is controlled by motor neurons of the nervous system. So motor neurons are voluntary signals sent from the brain to the muscle, telling the muscle to contract. And so it initiates a neural impulse in the muscle tissue. What happens is that it causes a rise in calcium. That triggers a complex series of reactions. And then the myofilaments begin to slide over one another, and the muscle contracts or shortens. Every contraction needs a new stimulus. So calcium is actually more important to muscles in the muscular system than it is for the skeleton, at least in the body's eyes. Because if you are lacking calcium, your body will actually take it out of your bones so that your muscles can use it. So you do not want to have a calcium deficit in your body because it's going to cause a weakening of the skeleton because your body will then leach it out to be able to give to the muscles. So more specifically about the myosin are the thick filaments that are found in the middle of the sarcomere. They have a rod-like tail and two globular heads. So the myosin that we find in the middle of the sarcomere is actually many myosin filaments all interwoven together. The actin, remember, are those thin filaments that are attached to the Z lines. And when the muscle is contracting, the myosin heads will attach to active sites on the actin during contraction. And then the myosin heads will end up pulling on the actin, which then will initiate the shortening of the muscle. So here is a more close-up arrangement of a sarcomere where we can see the thick myosin filament in the middle with those globular heads, and we can see the thin actin filaments off the sides. Now in a relaxed muscle, the actin and the myosin overlap only slightly, and there's no binding. The globular heads are not attached to active sites on the actin. In a contracted muscle, the myosin heads start to bind to the actin active sites asynchronously. What that means is they're not all going to attach and pull at the same time. Each of those globular heads are going to be doing that at separate times. And you can see that in the animation on the bottom right of the screen. When the myosin heads bind to the actin, that binding creates something called a cross bridge. and then the myosin head will pull the actin towards the M band. It'll pull it towards the center. The myosin heads will bind and detach several times during a sustained contraction. So they'll bind, pull, continually. The actin will slide past the myosin, the sarcomere will shorten, the H zone disappears, the I band shortens, the Z discs become closer together, and this requires a great deal of ATP, which is adenosine triphosphate, or the usable energy produced by the mitochondria in our bodies. So you can see on the image to the left a comparison between a relaxed and a contracted sarcomere. And then on the right, you can see a conceptual diagram of a sarcomere that might help you visualize it a little bit more. Person M, which is the myosin, is standing between two bookcases, which are the Z bands, and pulls them together via the ropes, which is the actin. So when when the myosin is grabbing onto the actin, like we see in that picture, that would be a cross bridge. And it's going to pull those Z bands or Z lines towards the center. So specifically, there are 4 sequential events of contraction. First is cross bridge formation, and that's where the myosin attaches to the actin, forming the cross bridges. 2 is the working or the power stroke, and that's when the myosin pulls the actin toward the center of the sarcomere. 3 is cross bridge detachment, and that's actually what the ATP is used for. And then 4 is the cocking, or going to the original position of the myosin head. The head pivots back to the original state so that it's ready to go through this sequence again by forming another cross bridge, working power stroke, cross bridge detachment, and so on. And so the animation on the bottom right of the screen, you can see that sequential event happening over and over. So some contraction facts: a single power stroke, so one movement of one myosin, will shorten the muscle approximately 1%. Muscles, on average, shorten to about 70% of their resting length. And on average, muscles can contract at a rate of 8 meters per second. So all of this happens very, very quickly. Now, after death, there are some very specific stages that occur with the body, and these have a lot to do with muscles. So the first stage after death is called livor mortis, and this is when blood will pool at the lowest part of the body, and this happens immediately after death. And so that's going to result in discoloration, where there's going to be darker coloration on the lower parts of the body. Second stage is algor mortis, and this is when the body temperature drops. This starts 30 to 60 minutes after death and lasts a few hours. And then finally, rigor mortis is post-mortem rigidity. This starts a few hours after death, peaks at about 12 hours, and lasts up to about 48 hours. Now, with rigor mortis, this has a lot to do with muscle contraction and ATP, because what ends up happening is the muscles, the myosin and the actin will form those cross bridges, and the power stroke will take place. But because death has occurred, the body is not going to be producing any ATP. And so after that cross bridge is formed, there's no way for it to detach. And when that starts to happen with all the myosin and all the actin of all the muscles, that's where we get the stiffness. Now, when rigor mortis starts to subside, that's due to protein decomposition, where the muscles are actually starting to go through the decomposition process and break down, which is why those cross bridges start to break. So rigor mortis is when huge quantities of calcium are released upon death. It allows myosin heads to attach and pull on the actin, but no ATP will be produced to break the cross bridges, and it subsides due to muscle decomposition. A motor unit is the neuron and all the muscle fibers it connects to. There are many motor units connected to each muscle, and they're not localized, they're spread throughout the muscle. And so this is what's going to cause individual muscle fibers or muscle cells to. The strength of contraction depends upon the number of motor units that are stimulated in muscle. So small motor units connect to fewer fibers. Because of that, they produce less power upon contraction, but they're also in control of more fine and precise movements. Where large motor units will connect to more fibers. Because of this, the contraction will have more power, but it means it'll have less precise movements. And anytime we want a stronger contraction, we have to recruit or stimulate more motor units. The all or none response is the strength of response of a muscle fiber, and it's not dependent upon the strength of the stimulus. So once a muscle fiber is stimulated by a motor neuron, the muscle fiber is fully committed to a full contraction. So it's not like we can send a weak stimulus or a strong stimulus. A stimulus will cause that muscle fiber to fully contract. So what happens is actually recruitment. Recruitment is the activation of additional motor units to increase contractile strength. The more cells contract, the stronger contraction. And so it has nothing to do with the strength of the stimulus. The strength of contraction has everything to do with how many muscle fibers are being told to contract. So the image on the top is showing a single motor unit that's connected to 5 different muscle fibers. So when that motor neuron has a message to contract, all 5 of them will contract. Where picture B is showing 2 motor units. We have the blue neuron controlling 4 muscle cells, and we've got the red neuron controlling 3 muscle cells. We could have the blue neuron stimulate the muscle cells, and then 4 of them would contract. We could have the red one stimulate its muscle fibers, and then 3 would contract. That would actually be a weaker contraction because fewer muscle fibers are being stimulated. And if we wanted a very strong contraction, we would tell both motor neurons to cause their cells to contract. If we look at this example, motor neuron 1 would produce half the force as motor neuron 2. Smaller motor units allow more graded force production. Larger motor units allow more overall force. And here is an example of recruitment. How with one unit recruited, we have a weaker contraction. If we recruit 2 units, we're going to have a stronger contraction. And 3 units, we're going to have an even stronger, faster contraction. And you can see that both by the graph as well as the animation in the upper left corner. Another analogy for motor units, if you want more light, you turn on more than one lighting unit. Similar with muscles, if you want more muscle tension, you turn on more than one motor unit. So as we increase the number of stimuli, we're going to increase the strength of the contraction. And so one single stimulus is called a muscle twitch. And a muscle twitch actually has 3 different periods to it. The latent period is when the stimulus is first received, which stimulates the release of all this calcium. The second period is the contraction period, and that's when the muscle actually shortens. And then we've got the longer relaxation period, which is when the calcium clears from the area and the muscle returns to its original length. Muscle twitch, we don't normally observe in a normal muscle, but this can be provoked in a lab setting with an electromyograph, an EMG, or a nerve conduction velocity test, which will measure muscle response to nervous stimulation. So if we increase the stimuli with shorter relaxation periods, we do not give enough time for that calcium to fully clear, and so it allows the contraction to intensify in strength. So wave summation is when a second stimulus is applied before the muscle is completely relaxed, and that induces another contraction that adds to the first. So the successive stimuli induces a summed contraction tension. Unfused tetanus is when the stimulus frequency increases, so the tension rises to a fairly steady value. So the individual twitch contributions are still seen as bumps on a myograph because we're still seeing the relaxation period start before the next stimulus is given. Now, fused tetanus is how our muscles normally function. This is when there's even higher rates of stimulation that results in no significant or no sign of individual twitches on a myogram. Now, each of those, you can see wave summation, you're going to have about 10 to 40 stimuli per second. Unfused tetanus, you're going to have about 50 stimuli per second. And then with fused tetanus, our normal muscle function, you're going to have greater than 60 stimuli per second. And here is a graph that's just showing all 4 of those together. Moving on to exercise. For exercise, we have something called treppy. And treppy is the warm-up period, or it's sometimes called the staircase effect. And this is when there's a series of increasingly stronger contractions after identical stimuli is applied to a rested muscle. And it's up to 2 times increased strength of contraction after multiple stimuli of the same strength. Now, this is different than what we were just talking about with wave summation and unfused and fused tetanus, because with those, we were not letting the muscle finish its relaxation period. And here, the muscle gets to fully contract, and then another stimulus is given. And because of that, eventually the muscle will have stronger contractions. The reason this happens physiologically is because more calcium is available. Since we've been using the muscle, there's calcium readily available for contraction, and the enzymes that are used in the chemical reactions for muscle contraction become more efficient as heat is released. And remember, heat production is one of the functions of muscle action. So here is that comparison. Treppy is an increase in peak tension with each successive stimulus delivered shortly after the completion of the relaxation phase. The fiber's maximum potential tension is not reached until tetanus. Where in wave summation, it occurs when successive stimuli arrive before the relaxation phase. So in treppy, each successive stimulus is administered after complete relaxation, and in wave summation, the muscle is not relaxed prior to each successive stimulus. More on exercise. There's isometric versus isotonic contractions. In an isometric contraction, like the image on the top, it increases muscle tension, but the muscle stays the same length. There's no movement produced. And so that would be like doing that sort of action as you see in the picture on the top, where there's definitely tension in the muscles, but we're not shortening or moving anything. Isotonic contractions, on the other hand, will decrease muscle length and cause movement. So that's the easiest way to determine if something's isometric versus isotonic. Is there no movement or is there movement? Now, if we look further into isotonic contractions, they can be divided into concentric and eccentric. A concentric contraction is when the muscle shortens and two connection points come together. And that's what you typically think of when you think of exercise. You think of using your muscle, shortening your muscle, and that. Eccentric contraction is when the muscle lengthens. It's when there's two connection points that are moving further apart. This acts as a braking system. It prevents the weight from dropping too quickly due to gravity. So you're still contracting, but it's slower and it's actually going to result in lengthening. So here's those three types compared side by side. So we already know that isometric, there's not going to be any movement. That, for example, that's when you're holding a weight. The bicep stays the same length, but you are definitely building tension in that muscle. So the two isotonic contractions, concentric is like the upward phase of the bicep. The bicep shortens and pulls the forearm upward. Eccentric would be the downward phase because the bicep is still producing some tension while it lengthens. It slows the lowering of the forearm and controls extension of the elbow. So even though most of the contraction is with the concentric movement, the eccentric movement is still going to show some tension to help control the movement. And here's just some more examples. Endurance exercise is also called aerobic. These are things like swimming, jogging, biking, activities that require moderately weak but sustained activity for longer periods of time. This actually changes the muscle physiology. It increases the number of capillaries and mitochondria within the muscle fibers. This creates more efficient oxygen use and muscle metabolism. You have greater resistance to fatigue, but it does not promote muscle hypertrophy. And what that means is it's not going to build and bulk up the muscles. It's going to change how the muscle functions, but it's not going to increase its size. This relies on and promotes the development of something called slow twitch muscles. Slow twitch muscles contract slowly and work longer without running out of energy, mainly because it has that extensive blood supply because of increased capillaries and increased energy supply due to excess mitochondria. Resistance or anaerobic would be activities like sprinting, weightlifting, isometric activities. It requires high intensity bursts of strength, but for short periods of time. This also will change the muscle structure and physiology. It increases the number of myofilaments. It increases the thickness of the endomysium, and it increases glycogen stores. And because of that, this does result in hypertrophy because the muscle fiber itself is going to increase in size. And if all the muscle fibers increase in size, you're going to have larger muscles. The glycogen supplies quick energy stores. This relies on and promotes development of fast twitch muscles. Fast twitch muscles are stronger, they contract quickly, but they tire much faster. And they don't use oxygen as efficiently. Instead, they're functioning without oxygen, which produces a byproduct called lactic acid. And that's what causes the burning sensation that we sometimes feel in our muscles. Now, keep in mind, our muscles do not get larger because our cells are dividing. Our muscle cells rarely divide. So any increase in muscle size is going to come from increased size of those individual fibers, not additional fibers being produced. So this is just a comparison of those slow twitch and fast twitch. Now, every muscle has a combination of slow and fast twitch fibers. The average person has approximately 60% fast twitch fibers and about 40% low twitch fibers. Each individual is genetically predisposed to a certain amount. So genetics is going to determine, in general, how much fast twitch and how much slow twitch fibers your muscles will contain to a certain extent. So you do not gain or lose muscle fibers due to training or lack thereof. Various types of exercises bring about changes in skeletal muscle fibers. Like we said, how the endurance exercises can cause increases in capillaries and mitochondria. How anaerobic resistance exercises can result in increased myofilaments and increased endomysium, which actually causes muscle to get larger. So endurance type exercises will transform fast into slow fibers. Endurance athletes have anywhere between 70 to 80% slow fibers. Those would be your marathoners or your triathletes. So you definitely can bring about a change. Just keep in mind, though, that there is a genetic predisposition. So your body has like a range of what it is able to do that change for. Resistance exercises will transform slow into fast fibers. This results in hypertrophy because of increased cell size, not increased number of cells. And power-based athletes can have about 70% fast fibers. This is your sprinters or your Olympic weightlifters. Now, lack of exercise can do the opposite. Lack of exercise can result in atrophy, where the muscles start to wither, and that's due to decreased cell size, not number of cells. So it can go either way. You can be building up your muscles and causing positive changes within the cells. You can also be creating negative changes in the cells with lack of exercise. So endurance versus resistance versus atrophy. Metabolic muscle fatigue. Muscle fatigue is officially the inability to contract even though stimuli is being sent and received. This results in weakness and a decline in the ability of the muscle to generate force. It results because there's depletion of molecules that facilitate contraction, things like depleted calcium, depleted ATP, and the accumulation of molecules, which impair contraction. So, byproducts that can actually get in the way of muscle function. So, lack of ATP availability, this is, as we talked about, kind of with rigor mortis, where the muscle remains shortened, but that ATP is not available to break those cross bridges. And so it leads to contractures or cramping. Lactic acid accumulation due to lack of oxygen. So if you are lacking oxygen for your mitochondria to produce energy, you're going to start to producing energy in an anaerobic or without oxygen way, which results in lactic acid. Lactic acid actually alters the pH balance in your muscles and makes it more acidic, which inhibits how well the muscle functions because that's changing the pH of the enzyme systems. Potassium builds up as a byproduct of muscle action. So as you use your muscles, potassium starts to build up. That alters the ion balance and it actually prevents calcium from being released. So those are three things that can lead to muscle fatigue.